In part 3 of our series on innate immunity, we're going to focus on the role of endothelial cell activation in mediating the transmigration of leukocytes from circulation into inflamed tissues. So we've talked a lot about how pathogens are recognized by cells of the innate immune system and how pathogens activate the complement cascade. Now we're going to turn our attention to how cytokines that are produced and how complement components that are induced lead to the activation of endothelial cells and a cellular infiltration into inflamed tissues. There are a number of cytokines and inflammatory components that can mediate activation of endothelial cells and their vasodilation. The key ones that I want you to know about are the complement components C3a and C5a, as well as the cytokines interleukin-1 and tumor necrosis factor. These cytokines induce the vasodilation of endothelial cells, which leads to gaps in the tight junctions of local capillaries. Now, it turns out that if we look at these local capillaries and we look in the lumen, they are absolutely full of expression of cell surface proteins that are heavily glycosylated, which leads to the formation of this glycocalyx. And so I love this slide because it really shows in fine detail in this electron micrograph the formation of this glycocalyx on the lumen of the endothelial cells. Inflammatory cytokines increase the expression of a number of molecules that make up this glycocalyx, so it becomes stickier and thicker in activated endothelial cells that are exposed to inflammatory cytokines. Now, there are a number of molecules within this glycocalyx, such as addresins and selectins and integrins and immunoglobulin-like molecules. All of these molecules can mediate trafficking of immune cells from the lumen of the capillary into tissues. And again, I've just shown you this slide so you can see the many different types of molecules that are present at the surface, each with unique functions, but you don't have to memorize these. Instead, I'm going to ask you to focus on one of these integrins called LFA1. So how does LFA1 work? So let's look at a macrophage that gets activated. It produces some inflammatory cytokines and it also produces chemokines such as interleukin 8. It produces complement components such as C3a and C5a, and these inflammatory cytokines and these complement components activate local capillaries. When this happens, these local endothelial cells start to produce their own chemokines, and in this case, they're producing this chemokine interleukin 8. Neutrophils are constantly rolling along these capillary beds, along that glycocalyx layer, and when they bind to these chemokines that are being produced in this localized area, it induces their ability to express this integrin called LFA1, or leukocyte factor for adhesion 1. And when LFA1 is expressed, it mediates the firm adhesion of these rolling neutrophils or rolling leukocytes to the capillary. They essentially stop and they start to squeeze in between these tight junctions into the tissue. This is a process that we call transmigration. You will see it called other things from time to time, including diapedesis or extravasation, and they all mean the same thing. I'm going to use exclusively the term transmigration to describe this process. Once these cells are in the tissue, they can then migrate along a chemokine gradient from the site of the capillary to the actual site where the pathogen is being recognized by macrophages. And so this migration then leads to localization to the actual site of injury and pathogen location. Now, many leukocytes, not just neutrophils, use very similar mechanisms to migrate into inflamed tissues, but the chemokines may differ. The number of chemokines that are known is vast. Again, I'm not asking you to memorize a bunch of different chemokines. We are going to use the chemokine interleukin 8 as our kind of canonical example chemokine. In the next lecture, we'll discuss what those cells of the innate immune system do once they get into those tissues to eliminate the pathogen.